Hey, listeners, and welcome to episode 118 of Cell and Gene, the podcast. I'm your host, Erin Harris. My guest for this episode is Alexander Cryer, Ph.D., instructor in medicine at Mass General Brigham. Alex dives into a truly creative approach to cancer immunotherapy during our chat. He and his team have been exploring how to turn tumor cells from immunosuppressive bystanders into active drivers of anti-tumor immunity by harnessing the cGAS-STING pathway and mRNA lipid nanoparticle delivery. Alex walks us through the science behind reprogramming cancer cells to produce immune activating signals, the technical challenges of targeted nucleic acid delivery, and how this strategy could enhance existing treatments and even inform future vaccine and infectious disease applications. We cover so much more in probably what is one of our most fascinating episodes yet, so be sure to tune in. And so I'm going to let you go and listen in. Here's my conversation with Mass General Brigham's Dr. Alex Cryer. Enjoy. Alex, hi. Thanks for joining me on Cell and Gene, the podcast. I'm happy you're here.
Thank you very much for the invitation. Very happy to be here as well.
Good. So talk to us a little bit about the research that you and your team did. So you and your team are with Mass General Brigham, and you explored the idea of harnessing tumor cells' own machinery to promote, anti-tumor immunity. Talk to us a little bit about the research, where we are today, and how does this change our traditional approach to cancer immunotherapy?
Yeah, sure. So the idea was actually generated whilst I was working on a different project, believe it or not. So when I first entered the lab, I work in the lab of Dr. Natalie Artsy here at MGB, and at the time, and still now to some degree, we were interested in delivering innate immune agonists, and we were working on delivering a nanoparticle formulation of an innate immune agonist, specifically a STING agonist. And as I was kind of acquainting myself with the field, kind of learning, because this was kind of my first entry point into this field at this point in my career, I started sort of getting a picture of a couple of things in this field that were quite striking. And some of the things that struck me were that the cGAS-STING signaling axis that exists in all mammalian cells and indeed other cell types as well, the species, it was typically dysfunctional in cancer cells. And we can discuss a little bit more about what that means a bit later potentially, but it wasn't working in the appropriate fashion in cancer cells. And in some cases, they'd actually hijacked this pathway for their own benefit, which was quite an interesting discovery. And then another element that I discovered as well in the literature is that the molecule that is produced in this pathway endogenously in mammals is called cGAMP. This is produced by cGAS, and cGAS is an enzyme. This is produced also inside the cancer cells, but the cancer cells and other cell types too seem to transfer or export this molecule outside of the cell because it acts intracellularly. So it seems like the cancer cells don't really want this molecule to be around very much. And so, and then when it's outside of the cell, it gets degraded really quickly. And so kind of looking at these two things, I was like, okay, well, maybe there's some vulnerability there we could potentially exploit. And as you mentioned in the question, that typically a lot of focus is on the immune cells in immunotherapy. And indeed, our approach with the nanoparticle STING agonist formulation was very much immune cell focused as well. But then I thought to myself, well, in the tumor microenvironment, what cell type actually comprises the most of that tumor microenvironment? It is the tumor cells, typically in almost every tumor. And so I thought, well, okay, there's this potential vulnerability that could be exploited. And we figured that potentially we could try to leverage some element of this pathway in order for these cancer cells to produce more of this molecule they don't want around. And because there are so many tumor cells compared to immune cells, maybe this could be another interesting strategy to kind of get the cancer cells to produce a molecule that could actually activate the immune cells in the surrounding TME instead of relying on the very scarce immune cells that populate the TME typically in tumors. And so this is kind of how we arrived at this strategy in a sense to try to basically leverage the numerous tumor cells in the microenvironment to produce this immune activating molecule.
Thank you for that. Excuse me. Okay, so you mentioned cGAS-STING and cGAMP, of course. Talk us through how cGAS-STING, that pathway, was restored in tumor cells, and perhaps how boosting cGAMP, production leads to broader immune activation within the tumor microenvironment.
Yeah, so one of the really nice things about this is that evolution has done a lot of the work for us. And so we were basically trying to sort of hyperactivate this pathway transiently and temporarily. And so what happens typically in viral infections, this is the kind of the backdrop with which this pathway was sort of discovered in is that cGAS is an enzyme and it detects double-stranded DNA in the cytoplasm. And that can be from self or it can be from foreign. So some viruses have double-stranded DNA, for example, and this can be detected by cGAS as well. And so when cGAS detects intracellular double-stranded DNA, it then produces this molecule cGAMP, which is a cyclic dinucleotide. And this molecule is a STING agonist. It's one of the mammalian endogenous STING agonists. And so this molecule is produced in the cytoplasm upon cGAS sensing double-stranded DNA, and then cGAMP will be produced and then activate STING. And so where our kind of strategy is reliant is that in cancer cells, if we, for example, were to increase the production of cGAS, we would get more cGAMP, as cancer cells have a proclivity to have much more double-stranded DNA floating around in the cytoplasm than healthy cells. And so the idea was that we kind of transiently increase cGAS expression in these cells. This would produce more cGAMP. And as I mentioned in the first question, there's a tendency for these tumor cells to export cGAMP outside of the cell. And so we thought we could, again, leverage this process to get the cancer cells to export lots and lots of the cGAMP molecules to the outside of the tumor cells and into the tumor microenvironment. And there are lots of immune cells. And it's been discovered that there are certain importers of these cyclic dinucleotide molecules on immune cells and other cells as well. So we were trying to basically get the cancer cells to produce all of the cGAMP, which would then be imported into the immune cells. And I mentioned earlier that the cGAS-STING pathway in tumor cells is typically dysfunctional in some way, shape, or form. But in immune cells, it generally is more competent. And so the immune cells would have this active STING. And this is what we were really relying on to basically promote and instigate anti-tumor activity. So the cGAMP would be imported into the immune cells. It would then activate the STING in the immune cells. And we and many others have shown that the host STING and not the cancer cell STING is vitally important for an anti-tumor effect. And then upon STING activation in these immune cells, you would get the production of type 1 interferons and many other pro-inflammatory cytokines and chemokines, which have been typified as these molecules that are important for instigating this anti-tumor response.
Great, thank you. Now, this approach involved mRNA lipid nanoparticle delivery to reprogram the cancer cells. So, what were the largest technical challenges in applying this strategy? And then how did you overcome them in your preclinical models?
Yeah, so I would say there were two principal technical difficulties when trying to apply the strategy. The first one is just delivery of nucleic acids in general. This is a barrier that many scientists and people around the world are familiar with. And the second one was trying to achieve this kind of tumor targeting or selectivity. And so this was a proof of concept study, and we wanted to sort of just try to see if our hypothesis had any grounding whatsoever. And so to do that, to overcome the first barrier, the delivery barrier, this is where, as you say, we implemented lipid nanoparticles. And so I'm very fortunate to be in a lab that has a lot of nucleic acid delivery knowledge and a toolkit. And I've been here for a few years too, so I've learned a thing or two. And so working with colleagues in the lab and also some literature precedent, we were able to develop a lipid nanoparticle formulation that was readily able to encapsulate our custom mRNA for cGAS and also to deliver it really effectively to tumor cells. And so this is kind of how we overcame the delivery element of that problem. And then in terms of the tumor targeting and selectivity, this is a perennial kind of issue in the drug delivery field of how do you achieve cell and organ specific targeting. And indeed, we've not cracked that code necessarily here, but what we did to overcome that was we adopted intratumoral delivery. So we kind of cut out the problems of systemic delivery and other delivery routes as well, and we just kind of went straight for the target. And preclinical models, I mean, for their faults and weaknesses, they do enable you to try these things in a relatively fast and sort of resource inexpensive setting. And so we did intratumoral injection of these lipid nanoparticles directly into the tumor, so that it had the maximum chance of transfecting the cancer cells and for us to actually test our goal, because shifting away from that would add in difficulties and other barriers that would maybe muddy the waters for us actually answering the question that we were interested in.
Wonderful, thank you. So We know that your study combined cGAS restoration therapy with immune checkpoint blockade, and you saw enhanced outcomes. So, how do you see your findings informing future combination therapies for tumors that traditionally resist immunotherapies?
Yeah, that's a really good question. So just want to make a point that first, we're obviously really happy to see that we did see some added benefit of the immune checkpoint blockade in our study. It is relatively well known in the field that STING agonists do combine well with immune checkpoint blockade in preclinical models, and so we were glad to see that our other strategy could also fit into that narrative, and we were able to contribute to the literature in that way too to further cement that finding, which was nice. Um, I would say the main area for opportunity in the therapeutic approach that we took was you really want to, from my understanding, get more double-stranded DNA. The more that you have, the better in principle the response would be, because you would produce more cGAMP, you would be able to then activate more STING, more type 1 interferon, etc. And so um, somewhat interestingly, there are clinical modalities available currently, such as radiotherapy and certain kinds of chemotherapy that can induce double-stranded DNA in the cytoplasm just by the nature of how they work. They are kind of DNA interacting agents in some way, and they cause double-stranded DNA breaks and all sorts of DNA damage. And so I think the area where this approach could be potentially most immediately therapeutically beneficial would be in combination with things like radiotherapy and certain kinds of chemotherapy. There are also other agents that can induce certain immunogenic cell death pathways that, again, can release these kind of double-stranded DNA fragments. And so I think that area would be a really, really interesting area to explore in the future, something that we're definitely interested in looking into.
Good. Okay. Now, speaking of areas we're going to be looking into, beyond cancer, your paper suggests potential applications for vaccines. What are the next translational steps for this approach?
Yeah, also a fantastic question, something we've been thinking about quite a lot. So just to touch on the vaccines comment, a paper from Professor Jonathan Kagan really nicely showed that catalytically active cGAS mRNA delivered with antigenic mRNA was able to induce better vaccine responses in preclinical models. So this is kind of one of the areas where it can be applied in the vaccine space. Indeed, the mRNA that we used was not catalytically active in and of itself. It had to become activated when it was translated. So I think if you're able to use that strategy, that's a really good direction forward. I know that thinking beyond cancer was one of the scopes of the questions, but of course we only tested it in one type of tumor model, and there are different types of tumor models we'd be interested in looking at as well. There are some that are more or less sensitive to radiation or chemotherapy, and as I mentioned in the kind of previous segment, I think that's a really interesting area of combination therapy to look at. So there are other tumor models that could be potentially interesting to look at as well to see if we see that synergy that we would hope to see. And I think if we're able to get a catalytically active version of it, or to provide a double-stranded DNA substrate for this enzyme in some way, there's definitely applications in infectious disease as well. I believe where that type 1 interferon response is definitely implicated in eradicating viral infections. So I suspect that a combination of those areas is probably where this therapeutic approach would go. Again, there would be developments that would be needed, and of course, adapting it away from intratumoral delivery to other delivery routes. If we're able to get an intravenous friendly delivery system, especially with an element of targeting, perhaps, again, if you want to go for respiratory infections, if there's a way to get a lung targeting formulation of this mRNA there. Something that our lab is working on is looking at different targeting modalities for nucleic acid delivery. This would be something I think would be really interesting to pursue as well. I just want to sort of share that and kind of reiterate that this was a proof of concept study, but we were really excited about the results, that kind of something that you've kind of have pieced together from the literature and the hypothesis that you generate over a period of years kind of comes together in this work, which was really exciting. And that the cancer cells, because they are so numerous, they're not just malignant bystanders in a sense. Obviously, they contribute tremendously to tumor progression, not just within themselves, but the other cells around them. But there is, I think, a way to start thinking about how do we actually tap into that really numerous cell population from a therapeutic perspective. This is something that I think one of the essences of the messages I wanted to kind of convey with this work, and that evolution has done a lot for us in terms of providing these really powerful innate immune signaling pathways, and the immune system in general is an extremely powerful and complicated tool. I think if we're able to kind of leverage those evolutionarily kind of instilled mechanisms in our physiology, this is a really interesting and elegant way to try to kind of think about how to better treat the plethora of diseases that we have way beyond cancer into every other kind of disease area.
Thank you. Thank you for that. All right. Well, that brings us to the end of our episode for Cell and Gene, the podcast. And I want to sincerely thank Alex Cryer, PhD, instructor in medicine at Mass General Brigham, for his time today. Alex, thank you so much for being here. This was a really thoughtful episode, and we would love to have you back to talk to additional research and or the progression of this particular project.
Well, thank you very much for the invitation again. It was a pleasure to discuss this with you, and I'll be very happy to be a recurring guest on this podcast whenever I have some more interesting findings to share.
Wonderful, wonderful. And listeners, thank you for tuning in. Please subscribe to Cell and Gene, the podcast, wherever you podcast, and we will talk to you soon.